We've seen how steroid hormones and other signal molecules that can diffuse across membranes can deliver information directly into a cell. In this module, we look at the mechanisms that large and/or polar signal molecules, like hormones, growth factors, or cytokines, that can't cross the plasma membrane, use to deliver their chemical message to control a target cell's activities. Signal transduction produces many different effects in cells, from regulating enzyme activity to controlling gene expression. A large or polar signal molecule binds to the cell surface receptor, shown here. The cellular response includes the phosphorylation of proteins, and when activated by phosphorylation, these proteins are responsible for a variety of different effects, ranging from, again, changing the activities of still other enzymes, to rearranging cell shape by manipulating the cytoskeleton, to increasing or decreasing ion permeability of cellular membranes, to turning genes on and off, to promoting replication and cell division. This slide summarizes the themes of signal transduction. The signal molecule binds to a specific receptor on the outside surface of target cells, resulting in the activation of intracellular protein kinases. These are enzymes that catalyze the phosphorylation of other cellular proteins. To control how intensely and how long the cell responds to the signal, there are specific phosphatase enzymes that catalyze the hydrolysis of the phosphates off of the activated proteins when the response is no longer needed. Here from your textbook are three main pathways for signal transduction, officially defined as the process of getting an extracellular signal inside the cell when the signal molecule itself cannot cross the membrane. The first one is ion channel linked receptors, which are characteristic of neurotransmission, as we've already seen. In this module, we're going to focus on signal transduction by G-protein and enzyme-linked receptors. Let's compare the G-protein and enzyme-linked signal transduction pathways in an overview. We'll look at the details in a bit. G-protein signal transduction involves the intracellular generation of second messenger molecules, for example, cyclic AMP, while enzyme-linked signal transduction does not involve second messengers. Both usually involve a series of protein phosphorylations, commonly referred to as a phosphorylation cascade, that eventually leads to the appropriate cellular response, as illustrated in this figure from your textbook. Now let's look closely at how G-protein-linked signal transduction really works to produce the second messenger molecule. In this case, it'll be cyclic AMP in a target cell. Now let's take a closer look at how G-protein-linked signal transduction works to produce the second messenger molecule, cyclic AMP, in target cells. The G-protein is the 3-subunit protein in shades of yellow, alpha, beta, and gamma, initially bound to GDP. This protein is called a G-protein because it's either bound to GDP, or as we'll see in a moment, to GTP. When the signal molecule, here shown in blue, binds to the cell surface receptor, which is shown in green, the receptor changes shape and can bind to the G-protein. This binding causes an allosteric change in the G-protein, leading to GTP displacing the GDP. In turn, the GTP-bound alpha subunit dissociates from the rest of the G-protein. The GTP-bound alpha subunit now has an affinity for and binds to an inactive adenylyl cyclase enzyme, the pink membrane protein, changing its shape. Now active, the adenylyl cyclase catalyzes the formation of cyclic AMP from ATP by hydrolyzing the phosphate linkage between the alpha and beta phosphates on the ATP, and then creating a cyclic compound. Cyclic AMP is produced faster than it is degraded as long as the hormone remains bound to the receptor and the GTP-bound alpha subunit of the G-protein remains bound to the adenylyl cyclase. The target cell, in other words, will keep making cyclic AMP to sustain the cell response until levels of the signal molecules drop in the blood, as illustrated here in the animation. The hormone on the receptor came off, setting off a sequence of allosteric changes that are the reverse of what happened during signal transduction. The GTP-bound alpha subunit eventually dissociates from the adenylyl cyclase, rendering the enzyme inactive. The GTP is hydrolyzed to GDP, and the complete G-protein simply reforms. Here is how cyclic AMP is formed and destroyed. You can see the formation of the cyclic compound first, and then you can see phosphodiesterase catalyzing opening of the cyclic compound to leave behind just plain old AMP. The best known example of a cyclic AMP action in eukaryotic cells is the fight or flight response to adrenaline, a hormone so polar it cannot get into target cells, in this case liver cells. After adrenaline binds its receptor, the G-protein mediated response, not shown in detail here, activates adenylyl cyclase, which results in cyclic AMP synthesis, which is shown here. Next, two molecules of cyclic AMP bind to an inactive protein kinase, a 4-subunit protein, which then dissociates. The cyclic AMP-bound subunits, shown in yellow in this cartoon, are not the active enzymes, but the other subunits, shown in purple that have come apart from the tetramer, are now each active protein kinases that can catalyze phosphate transfer from ATP to some target protein, which, as you can see, they do. The target in this case is called glycogen phosphorylase kinase, because when activated, it will catalyze phosphate transfer from an ATP to an inactive enzyme called glycogen phosphorylase, shown here. You may recall this as the first enzymatic activity in the breakdown of glycogen. The active glycogen phosphorylase catalyzes the hydrolysis of glycogen to glucose 1-phosphate monomers in the liver. You may also recall that glucose 1-phosphate is converted to glucose 6-phosphate, which would become dephosphorylated so that glucose can leave the liver cells and circulate to the brain or heart or other cells to enable fight or flight. One last note. The series of phosphorylations is called a phosphorylation cascade, more accurately a phosphorylation amplification cascade, because a small amount of starting catalyst, the protein kinase A here, can produce a large amount of active glycogen phosphorylase at the end of the cascade. This is, of course, the very nature of enzymes. Cyclic AMP is one of several second messengers in cells made during a G-protein mediated signal transduction. Some of the others are shown in this slide. First, let's include the cyclic AMP-mediated activation of protein kinase A, or pKa for short. Now let's look at what happens when the signal binds to the receptor and activates a membrane protein called phospholipase C. This generates two intracellular second messenger molecules. One is inositol phosphate 3, or IP3, shown here in the cytoplasm, and the other is diacylglycerol, or DAG for short, which is actually embedded in the plasma membrane. The IP3 causes the release of stored calcium ions from the smooth endoplasmic reticulum in the target cell, which in turn binds to a membrane-bound protein kinase called protein kinase C, or PKC for short. At the same time, diacylglycerol, or DAG, also binds to the PKC. With both DAG and calcium bound to the protein kinase C, this enzyme is fully active and initiates a phosphorylation amplification cascade, leading to its own ultimate cellular response. This slide simply summarizes many of the cyclic AMP and therefore protein kinase A-mediated cell responses, including changing plasma membrane permeability to specific molecules, remodeling the cytoskeleton to change cell shape, activating transcription factors in the nucleus to regulate gene expression, and even regulating protein synthesis on the rough endoplasmic reticulum, not to mention a few others. Protein kinase C and phospholipase C mediate some common response pathways, but also have some independent effects, noted on this slide. Specific examples of protein kinase C effects include the release of neurotransmitters from nerve cells, cellular differentiation, and participation in glycogen breakdown and fat synthesis in different tissues, as well as the secretion of hormones from endocrine glands, such as growth hormone and LH, which is luteinizing hormone, from the pituitary gland, and testosterone from the testes. These hormones are mitogenic, that is, they stimulate cell division in their own target tissues. Phospholipase effects include the stimulation of the release of hormones that enhance liver glycogen breakdown, and the secretion of amylase by the pancreas to help digest intestinal carbohydrates. Phospholipase C also stimulates liver production of some blood clotting factors, like thrombin shown here. Some chemicals mimic the mitogenic functions of PKC. One class of carcinogenic chemicals is the phorbol esters, normally found in some plants. As shown here, they can get into cells and bind to protein kinase C, activating it and causing the cell to respond as if it were responding to hormones that activate the protein kinase C. Because human cells have no way to get rid of the phorbol esters, the activated PKC has persistent mitogenic effects, leading to cancer and tumor formation. Most amino acids, or amino acid side chains, can't be phosphorylated. pKa and pKc are both called serine-threonine kinases, which just means that they catalyze the transfer of phosphate groups from an ATP to an appropriate serine or threonine side chain in the target proteins. Receptor tyrosine kinases are membrane receptors that have a hormone or other signal binding site on the external plasma membrane surface and a kinase activity in the cytoplasmic domain. Because they have their own phosphorylation activity, they don't need G proteins in order to transduce an external chemical or hormonal signal. The known receptor kinases all catalyze phosphorylation of target proteins not at serines or threonines, but at tyrosine side chains, hence the name receptor tyrosine kinase. This slide shows how receptor tyrosine kinases are activated when signal molecules, here called ligands, bind to the receptor domain. The word ligand is simply a generic term for any effector molecule that binds to a receptor. After ligand binding to monomeric membrane receptors, two ligand-bound monomers come together. This activates a kinase activity that phosphorylates each monomer at several tyrosines, as suggested here. When phosphorylated, the entire ligand or hormone receptor complex acts as a protein kinase to phosphorylate intracellular proteins, shown here a second ago, binding to the activated receptor, thereby transducing the signal information from the cell exterior into the interior of the cell and generating ultimately an appropriate cellular response. Here's a well-known example of how a receptor tyrosine kinase actually mediates a mitogenic response, that is, how it causes cells to start dividing. It begins with ligand binding, that is, a hormone, for example, binding and autophosphorylation of the dimerized receptor, as we've just seen a moment ago. The activated kinase domain recruits first an adapter protein from the cytoplasm, navy blue here, and then a protein called the RAS activating protein, shown in what, royal blue, I guess? In this state, a GDP-bound inactive membrane protein called RAS associates with the RAS activating protein, resulting in GTP displacing the GDP on the RAS protein. The RAS protein is now itself active. An inactive enzyme, oddly but appropriately as we'll see, named MAP kinase, then binds to the receptor RAS complex, as just happened in the animation, and itself becomes an active enzyme, and thereby an amplification cascade begins. Let's look at the rest of this cascade. Next, MAP kinase phosphorylates MAP kinase, which then phosphorylates MAP kinase, which in turn enters the nucleus and catalyzes the phosphorylation of a transcription factor that activates genes, ultimately leading to cell division, as illustrated in the animation that just blew by you here. You may wish to review this animation to help you digest what I just told you. The RAS gene was first discovered as a viral oncogene, delivered to normal cells by a virus and causing them to become cancerous. Only after its cancer-causing properties were pretty well known was this gene, this RAS gene, actually shown to be a normal gene in the human genome and as well in other animal genomes. In its normal context, the RAS gene is active at appropriate times, placing the protein in the appropriate target cell membranes. But when active in a virus-infected cell, the gene produces abnormal amounts of the RAS protein at times when it shouldn't be there, causing the cells that should not be dividing to start doing so. In other words, cells bypass normal controls on RAS function and maintain RAS GTP at abnormally high levels. One final note. Under normal circumstances, excess RAS activity is controlled by the protein MKP1, which just stands for MAP kinase phosphatase 1. We've already seen the role of a phosphatase in mitigating or terminating a hormone response. This is a gene that is activated as part of a normal mitogenic response, simply to prevent runaway cell divisions. An active oncogene, however, would exceed the cell's capacity to dephosphorylate mitogenic kinases, and that's why cancers develop. And that brings us to the end of this presentation.